structure of and functions of the immune system. The structure is mainly made up of a lymphoreticular system, which is a complex organization of cells of diverse morphology and many organs which are responsible for production of these cells. And when you come to the functions of the immune system, this based upon the type of immunity it produces, it can be classified into, the functions may be classified into either a humoral immunity, which is mainly given by the antibodies which are produced by the plasma cells, and the T cells, which mainly provide the cell mediated immunity. And the immune response is the response how the immune system will act towards an antigen. So coming to the structure of the immune system, the structure of the immune system is made up of immune organs and immune cells. The immune organs in turn are classified into primary and secondary immune organs, and the two classical examples of the primary immune organs are the thymus and bone marrow, and secondary immune organs are the spleen, lymph node, and MALT, which is called as the mucosa associated lymphoid tissue. And immune cells, the basic cells are the T cells and B cells, followed by null cells and the phagocytic cells. Then coming to the thymus. The thymus, you know, is an organ which develops from the third and fourth pharyngeal pouch, and this thymus is mainly situated in the lower end of the throat, which is just behind the sternum, and this develops well during the up to the pre-puberty level, and after which it starts decreasing in size and finally it atrophies. The main function of this thymus is it educates the pre-thymic lymphocytes to gain immunocompetence. Immunocompetence means the competency to attack an antigen. The lymphoid stem cells which passes through the thymus becomes immunocompetent by action of various hormones like thymolin. And the T cells, they migrate from the cortex to medulla. The thymus is differentiated into an outer layer which is called as the cortex and an inner layer to the medulla. So these immature T cells will pass via the cortex to the medulla, and as they cross from the cortex to the medulla, they gain many differentiating antigenic receptors. And these receptors are classified as cluster of differentiation, and they are called as CD markers. These CD markers are unique for each and every immune cells. And for example, this T cells, they acquire CD3 molecule and the T cell receptor in the thymus. So this is the main marker to identify that a cell is a T cell. Then coming to the bone marrow, it is equivalent to the bursa of Fabricius in birds, and pre-B cells functionally mature into the B cells, and then maturation and differentiation of pre-B cells is supported mainly by interleukin 7, and pre-B cells possess intracytoplasmic mu chains. Before migration to the peripheral lymphoid organs, this intracytoplasmic chains, they express the immunoglobulin M and immunoglobulin D on the surface. So that is one of the lineage markers for B cells. So this is the lymph node.
Excuse me, ma'am.
Pardon?
Ma'am, can you show the previous slide?
Previous slide.
Yes, ma'am.
Yes.
Ma'am, what is this bursa fabriculus in birds?
Bursa of Fabricius is similar to our bone marrow, which is present in the birds. So it was first identified in the birds that this bone marrow was responsible for production of these B cells, and these B cells will finally mature to plasma cells, and finally they produce the antibodies. So the bursa of Fabricius in birds, which was first identified, is equivalent to our bone marrow, which will mainly produce the B cells. Is it clear?
Yes, ma'am.
Okay. Now we'll move on to the next slide.
Okay, ma'am.
So now coming to the structure of the lymph node. The lymph nodes are usually placed along the course of the lymphatic vessels, which you very well know. They are surrounded by a fibrous capsule, and this fibrous capsule sends in trabeculae, which divides the lymph node structure into cortex, outer cortex, and an inner medulla. The node can be differentiated into an outer cortex and an inner medulla. In the cortex are accumulations of lymphocytes within which germinal centers are present. So these are the lymphocytes, and this white one is the germinal center. So what is this germinal center? The germinal center is the secondary lymphoid follicle, whereas the rest of the things are the primary lymphoid follicles. The secondary lymphoid follicles are formed in response to an antigen. Okay, and the follicles contain, besides the proliferating lymphocytes, they contain dendritic cells, macrophages, which are able to capture and process the antigen. And in the medulla, this is the medullary part. This yellow part is the medullary part. The lymphocytes, plasma cells, and macrophages, they are arranged as elongated branching bands, or they are also called as the medullary cords. The cortical follicles and the medullary cords contain B lymphocytes, and they constitute the bursa dependent area. So this lymph node has two different areas. One is the bursa dependent area, which will mainly produce the B cells, and another one is the T cell dependent area, which will mainly contain T cells, which are responsible for the cell mediated immunity. So the cortical follicles, these are the cortical follicles, and the medullary cords contain B cells, and they constitute the bursa dependent area. Between the cortical follicles and the medullary cords, there is an area called as the paracortical area. This pink color is the paracortical area, which consists abundantly of T lymphocytes, and this constitutes the T dependent, or the lymphocyte predominant area. So you have a B cell dependent area and T cell dependent area. The B cell dependent area is the follicles and the medullary sinuses. The T cell dependent area are the paracortical area, which is between the medulla and the cortex, which mainly consists of the T cells. Okay. Then coming to the spleen. The spleen is the largest of the lymphoid organs. It also has a capsule which sends in trabeculae inside, and it divides into interconnected compartments. The branches of the splenic artery travels along the trabeculae. They will travel along the trabeculae, and on leaving them, they branch again to form a central arteriole. So this is the central arteriole. This central arteriole is surrounded by a sheath of lymphoid tissue. So this white color is the central arteriole surrounded by the sheath of lymphoid tissue, which is called as the periarteriolar sheath. This part is known as the white pulp of the spleen. And inner to that, you have the red pulp of the spleen, which mainly consists of RBCs. And following antigenic stimulation, this white pulp of the spleen will mature to form mainly T lymphocytes, and these are the germinal centers which contain the activated lymphocytes. The periarteriolar lymphoid collection in the white pulp of the spleen are also called as the Malpighian corpuscles or follicles. Germinal centers develop. These are the germinal centers which develop following antigenic stimulation, and the lymphatic sheath immediately surrounding the central arteriole is the thymus dependent area of the spleen. So this white area is called as the thymus dependent area of the spleen. Okay, the spleen serves as the graveyard for the
IBC.
cells and platelets. So this is the main function of the spleen. So it is the largest of the lymphoid organs. So first we have finished the lymph node, and then the spleen, which are the secondary lymphoid organs. Then coming to the T cell and B cell dependent areas, which I have mentioned. The T cells are seated in certain areas which are called as the thymus dependent area. In the spleen, it is the white pulp or the periarteriolar sheath, and the paracortical area of the lymph node. Whereas the bursa dependent area is the mantle and germinal follicle of the perifollicular area of the spleen, and the cortical follicles are the medullary parts of the lymph node. Common MCQs usually asked in your examination: T cell dependent area and B cell dependent area. Then the other secondary lymphoid organ is the mucosa associated lymphoid tissue, which are subepithelial accumulations of lymphoid tissue in most of the lumen, like your respiratory tract, your intestinal tract, your genitourinary tract, etc. So it starts from the tonsils in the throat to the Peyer's patches in the small intestine. It contains a congregation of T cells, B cells, and the macrophages. That is about the lymphoid organs. And coming to the lymphosa, the cells mainly which comprise the immune cells are the leukocytes, which are classified into granular and agranular. Granular, you know, they are basophils, neutrophils, and eosinophils, and agranular are lymphocytes and monocytes. These lymphocytes are further differentiated into T cells when they pass through the thymus. When they differentiate in the bone marrow, they are called as the B cells, and NK cells are a separate entity of cells which are a part of innate immunity. So this is how the cells develop. T cells develop. First in the bone marrow, it is the hemopoietic stem cell which mature to the lymphoblast in the bone marrow. Once it reaches the blood, it forms the stem cells of the T cells, and then they are the immature T cells. These immature T cells pass through the thymus and they gain these receptors. CD3 molecule and T cell receptor are the two main markers for identifying a cell to be a T cell. This mature T cell finally matures either to have a CD4 molecule on its surface or a CD8 molecule. If it has a CD4 molecule on its surface, it is called as the T helper cell, and if it contains a CD8 molecule, it is called as a T cytotoxic cell or a T suppressor cell. So these are the markers. See, the main marker is CD3. The presence of CD3 indicates that it has passed through the thymus. And if it contains a T cell receptor, and if it contains a CD4 molecule, it is called as the T helper cell. If it contains a CD8 molecule, it is called as a T cytotoxic T cell. Then coming to the B cells produce antibodies. Apart from production of antibodies, the main function is it can also act as an antigen presenting cell. And on its surface, it contains cell surface immunoglobulin or the secretory immunoglobulin, class 2 MHC molecule, and C3b receptors. These B receptors is the complement receptors. And the two main CD markers for identification of B cells are CD19 and 20, apart from that 22 and 5. So this is the B cell maturation in the bone marrow. The pro B cell gets converted to pre B cell, finally to mature B cell. And this transition of B cell will finally mature to form the native mature B cells. So this, when they get activated, will form the plasma blast, and the plasma blast will now get converted or mature to a plasma cell, and these plasma cells are responsible for production of antibodies. This is how B cells mature. So the main markers of B cell are CD19, CD20, and the surface immunoglobulin. These three are the main markers to identify that a cell is a B cell. How do you differentiate T cells and B cells? Based upon certain. When you mix T cells and B cells with sheep erythrocytes, they form erythrocyte because of the presence of CD2 molecule on its surface, whereas B cells will not produce erythrocyte. Whereas when you mix sheep erythrocytes with an antibody and complement, forms the erythrocyte because the complement, B cells contain C3b receptors which activates the erythrocyte formation. And thymus specific antigen is CD3, which will be present in T cells, whereas B cells will not have CD3. And microvilli will be present in B cells, and that is the reason for the B cells which can act as an antigen presenting cell. This microvilli will engulf the antigen, take into it, process it and present it to the effector T cells. And blast transformation, that is conversion of a immature T cell to a blast one, is by means of concavalin and phytohemagglutinin for T cell, bacterial endotoxin and covan 1 Epstein-Barr virus for a B cell. And surface immunoglobulins are classical of B cells. And NK cells, as I told you, are called as the natural killer cells, which possess cytotoxicity towards the target. Cytotoxicity is not antibody mediated or MHC restricted. It is a part of innate immunity. It doesn't need any help from any other cell or etc. to get activated. It can activate on its own on exposure to an antigen. And they have two molecules, namely CD16 and CD56 on their surface. And they mainly target malignant and virus infected cells. And how do they destroy these malignant and virus infected cells? Is by means of production of perforins. Perforins are nothing but pores which extrude the cytoplasmic content and kill the cell. So this is the NK cell, which contains a CD16 molecule which will attack the tumor cells, form spores and finally cause lysis of the cell. So these are the markers. NK cell will contain CD16 and 56. B cell will contain CD19, CD20 and surface IgG, IgM and IgD. T suppressor or cytotoxic cell will contain CD3 and T cell receptor and CD8. The same way T helper cell will contain CD4. And next is the antigen presenting cell. What is the main function of this antigen presenting cell is they engulf the antigen, digest them into individual epitopes and present it to the effector T cells or the B cells. They are mainly found in the skin, lymph nodes, spleen and the thymus. The dendritic cells are mainly present in the B cell area of the lymph node and spleen, and all of them possess class 2 MHC antigen. But they themselves do not have antigenic activity, but they present the antigen to T cells, especially in case of primary immune response, and B cells in case of secondary immune response. And another classical example is your Langerhans cells, which can act as an antigen presenting cell. This is a dendritic cell which will contain fibrils which can engulf the antigen and present it to the effector T cells. Then coming to major histocompatibility complex, these antigen are cell surface antigens that induce an immune response leading to rejection of allografts. They are present on surface of all leukocytes, and their typing can be done whether it is MHC class 1, class 2 or class 3 by microtoxicity and mixed leukocyte reaction, and uses compatibility testing for renal transplantation. Any transplantation, MHC matching is a must. Disputed paternity, anthropological studies, because MHC molecules are peculiar to humans and other animals or birds, and association with certain diseases like HLA-B 27 as in case of rheumatoid arthritis. This HLA is present on the short term of chromosome 6, and they have three classes, class 1, 2 and 3. Class 1 is mainly responsible for the immune response, and they have contain A, B, C loci. And class 2 contains DP, DQ and DR loci. And class 3 are mainly the complement components. So this is class 1, which contains BC, class 2 which contains DP, DQ and DR, and class 3 is mainly made up of the complement components. So this MHC class 1 protein, they are made up of two chains, alpha and a beta 2 microglobulin chain. This beta chain is present on chromosome 16, and class 1 protein is present on virtually all nucleated cells. Can you name one non-nucleated cell?
RBC.
Very good. So all nucleated cells will contain class 1 protein, and they determine histocompatibility and acceptance or rejection of allograft. Whereas class 2 proteins are mainly responsible for regulating the immune response, and they are present only on cells of immune system like the macrophages, dendritic cells, activated T cells and B cells. whereas class 3 proteins are mainly components of the complement. So this is the class 1 protein, which contains an alpha chain and a beta 2 microglobulin. The alpha chain contains 3 domains, 1, 2, and 3, and the area between the alpha 1 and alpha 2 domain is the area where it is hyper variable, where the antigen present will come and get attached here. The same way in class 2, it has an alpha and beta chain, the alpha 1 and alpha 2 domains, and beta 1 and beta 2 domains, and the domain between alpha 1 and beta 1 is where the processed antigen will usually be presented to the MHC complex. And what is MHC restriction? See, there are certain types of cells which will get activated only when the antigen is in connection with one particular class of MHC. So T cells, they respond to processed antigens on macrophages and other accessory cells only when they are presented along with self MHC antigens, and this is called as MHC restriction. T cytotoxic cells, or the CD8 positive cells, recognize antigen when they are presented on MHC class 1, and T helper cells, or the CD4 positive T cells, will recognize antigens only with MHC class 2. This is called as MHC restriction. Is it clear? See, this is the antigen presenting cell. Class 1 antigen molecule with the antigen fragment, they have processed the antigen, will attach only to the CD8 positive T cell. Whereas MHC class 2, the antigen presenting only to helper T cell or the CD4 positive T cells. Then coming to immune response, based upon the cells involved, you have humoral immune response or the cell mediated. Humoral immune response is called so because mainly it is antibody mediated, and the antibodies are present in the blood, and they mainly are produce defense against extracellular bacterial pathogens, viruses of the respiratory and the GI tract, and they mediate type 1, type 2, and type 3 hypersensitivity. Whereas type 4 hypersensitivity is usually mediated by T cells and autoimmunity. So this is the humoral immune response. The antigen, once it is taken up by the macrophage or the antigen presenting cell, will be processed into minute epitopes. They will be presented with the class 2 MS to the helper T cells. These helper T cells will be proliferated in abundance, and then they will present the antigen or will stimulate the B cells. These B cells will finally mature to form the plasma cells, and these plasma cells will finally form the antibodies which can attack the antigens and destroy them. So this is how an humoral immune response takes place. Then coming to cell mediated immune response, it is the immune cells, namely the T cells, which take part, and they provide defense against intracellular bacterial pathogens, against fungi and viruses, rejection of graft, tumor immunity, type 5 hypersensitivity, and certain autoimmune diseases. So the activation phase, the antigen in class 1 MHC molecule, they usually present to cytotoxic cells, and then the cytotoxic cells are produced in abundance, which may in turn attack the antigen and kill them. This is cell mediated immunity. So how do you detect the presence of cell mediated antibody? You can detect by doing an antibody testing, which can be done in the present in the plasma or serum of the individual. But CMI, you can usually detect by means of delayed skin hypersensitivity testing, lymphocyte transformation test, target cell destruction, and migration inhibitant factor. So these are the 4 methods by which you can detect the cell mediated immunity. Then there are 2 types of immune response based upon whether the antigen is exposed for the first time or the second time. The primary immune response is usually slow, sluggish, and short-lived. You can remember 3 S: slow, sluggish, and short-lived, and they have a very long lag phase and very low titer of antibody, and the IgM which is produced is usually IgM. Whereas secondary immune response are 3 P's: prompt, powerful and prolonged, and they have a very negligible lag phase. As soon as the antigen enters the body, immediately the antibodies will be produced without any lag phase, and the amount of antibodies produced will also be increased, and the class of antibody produced will also switch over from IgM to IgG. So this is the primary response. About the lag phases, and the antibody is very minimal. Whereas when they are exposed for the second time, see, without any lag phase, they are increased, and the amount of antibodies is also produced in large quantities. And coming to cytokines, these cytokines are active substances which are liberated by activated lymphocytes. And cytokines are mainly peptide mediators that regulate the immune response. They may have paracrine action as well as an autocrine action. Paracrine action is mainly the lymphokine can act locally nearby the producing cells, whereas autocrine, the cells which produce themselves, can also be activated using these cytokines. And coming to the interleukins, there are nearly 13 different types of interleukins. We will see the most important one, interleukin 1, which is secreted by macrophages, monocytes, and other nucleated cells. It initiates proliferation of B cells, activate T cells to produce interleukin 2, and it produces fever. Once interleukin 1 is produced, only then we, as a part of inflammation, we produce fever, and that is why it is called as an endogenous pyrogen. Interleukin 2 is also called as the T cell growth factor because it is mainly produced by activated T cells, and it also activates the NK cells. And it usually converts the large granular lymphocytes to lymphokine activated killer cells, and they kill NK cell resistant tumor cells are usually killed by these lymphokine activated killer cells. And interleukin 3, they are produced by T cells, growth factor from bone marrow stem cells, and they are called as multi-CSF cells. Interleukin 4 are produced by T helper cells, and these T helper cells, this interleukin 4 produced by T helper cells, they activate the resting B cells, T cells, and mast cells, and they inhibit the production of interleukin 1. So they are all interrelated. One will suppress or activate the production of another interleukin.
Ma'am, what is the CSF?
Colony stimulating factor. Multi-colony, it will stimulate the whole lot of colonies of T cells and present in the bone marrow. Stem cells are the basic cells from which all the lineage cells are produced, like granular lymphocytes, granular lymphocytes, NK cells, etc. So these stem cells, it is called as a multi-colony stimulating factor. Okay.
Yes, ma'am.
Then interleukin 5 are produced by T helper cells. The proliferation of activated B cells and induce maturation of eosinophils. Interleukin 6, it will be difficult to remember, but no other go. Produced by T cells, macrophages, and fibroblasts. They act as an inflammatory response mediated in host defense. It also induces immunoglobulin synthesis in cells. It has stimulatory effect on hepatocytes, nerve cells, and hemopoietic cells, and they stimulate abnormal plasma cell clone, especially in multiple myeloma. So that is why in multiple myeloma, we usually look for interleukin 6. Okay. So they activate all T cells, macrophages, fibroblasts. They are produced by this, and they act as an inflammatory response. This interleukin 6 is one of a recent marker for identification of COVID-19 infection because it acts as a marker for inflammatory response against infection. So this interleukin 6, once it is identified to be increased in amount in an infected individual, he has more chances of lung injury. Okay. Then interferons. What are interferons? They are produced by virally infected cells, or they inhibit the translation of viral messenger RNA. That is the tip, which is also called as the transfer of the translation inhibition protein, and they are species specific. Say if adenovirus, all adenoviruses, this particular interferon will be and most viruses are better producers of interferons than DNA viruses. All interferons may be alpha, which are produced by leukocytes, and beta, which are produced by fibroblasts and epithelial cells, and gamma or immune interferons, which are mainly produced by the T cells in response to virus infection. And they have antiviral effect. Apart from that, the inhibition of cell growth, proliferation, and protein synthesis, and they increase the expression of MHC antigen. They also enhance NK cell, T cytotoxic cell, and macrophage activity. These two cells, because of the interferons, they mainly get activated to handle the virally infected T cells, virally infected cells. Then there is another factor called as the tumor necrosis factor, which are of two types, alpha and beta. And this alpha produced mainly by activated macrophages and monocytes, and tumor necrosis factor beta is produced by T helper cells. So the main function is they act as an acute phase response. They produce fever, macrophage activation, weight loss, vascular adhesion, interleukin 1 and 6 induction, and they produce the granulocyte monocyte colony stimulating factor induction. So it activates all the immune responses which are responsible to tackle an infection. Then there is another transfer factor. What do you mean by passive immunity? Anyone? Passive immunity? Shirisha? Kriti? Don't know. Passive immunity is transfer of ready-made antibodies. The same way, you can passively transfer cell-mediated immunity to an individual, and that product is called as the transfer factor. It is a polypeptide. It transfers cell-mediated immunity to all the antigen to which the donor is sensitive. It is usually used in T-cell deficiency, malignant melanoma, and in autoimmune diseases. And monoclonal antibodies, you know very well, which are produced against a single clone of cells directed against a single antigen. It is useful in diagnostic, therapeutic, and research fields. The antigen, once introduced into an animal, the immune cells are taken. These antibody forming cells of the B cells will be fused with the tumor cells. Why means this tumor cells has a capacity to proliferate innumerable amounts. So you will be able to produce innumerable amount of cells, which B cells, which will produce the specific antibody against this particular antigen. And that fusion of these two cells is called as the hybridoma. And these hybridomas are further stimulated to produce specific antibodies. And these are called as monoclonal antibodies. Monoclonal antibodies, they are helpful in treatment of various diseases. And did you read in the newspapers but that Trump was treated with the target bullet? Yes or no?
Yes, ma'am.
That target bullet is nothing but monoclonal antibodies against the coronavirus which was given to him. Okay. Then another group of antigens which you have to know is the super antigens. These are protein molecules which activate numerous T cells irrespective of antigenic specificity, and they mainly bind to the T cell receptor, and this exaggerated T cell activation results in outpouring of cytokines which leads to multi-system dysfunction. And the classical example of superantigen are the staphylococcal enterotoxin F, mycoplasma, and streptococci, which can act and stimulate the immune system and produce outpouring of innumerable number of cytokines, which can cause damage to the human system. So with this, we finish the theoretical part. We will move on to the questions. All of the following are characters of primary immune response. It is sluggish, except it is sluggish. It has a long lag phase, produces high titer of antibody. It does not persist for long period. So it produces high titer of antibody is the answer. Which of the following cytokine enhances proliferation of B cells? Is it interleukin 1, gamma interferon, interleukin 4, or interleukin 2?
Interleukin 1.
1.
Yes, ma'am.
Interleukin 1 is called as an endogenous pyrogen. It is interleukin 4 which stimulates the initiates the proliferation of B cells. Okay. It will be confusing now, but once you read and prepare a notes, you will remember. Peripheral lymphoid organs are centrally located in the abdomen to protect their vital functions, are designed to maximize contact between antigens, produce antigen-specific lymphocytes from stem cells in response to antigens, store large number of activated effector cells for a rapid response to the antigen, which is correct for a peripheral lymphoid organ. Anyone? It is D. It store large number of activated effector cells for a rapid response to the antigen. HLA typing is useful in this. You should answer. This you can't answer. Blood grouping, prognosis, cancer therapy, cases of paternal dispute. Which one? Cases of paternal dispute. Which of the following is an antigen presenting cell? Macrophages, dendritic cells, Langerhans cells, all.
All.
Good, all. A patient desperately needs a bone marrow transplant and a perfect match cannot be found. The rejection response is unmatched marrow is primarily due to the presence of mature T cells. You know very well, graft rejection is due to mature T cells that recognize the patient's recipient cells as foreign. To minimize this rejection, the marrow can be treated before transfusion into the recipient with the complement plus antibody to which molecule? In other words, which is the CD
CD8.
marker classical for T cells?
CD8.
CD?
8.
8.
CD4.
CD4. CD4 is T helper cell and CD8 is T cytotoxic T cells. But which is the marker that indicates it is the T cell? It is CD3. Okay. Surface immunoglobulins are present in immunoglobulins are produced by which cells?
B cells.
B cells. So B cells. A super antigen is best described by which of the following statement? It stimulates a large number of B cell clones. It is presented to a T cell after processing. It is viable and reproduces. It stimulates a large number of T cell clones.
Option D, ma'am.
D. D.
D. It stimulates a large number of T cell clones. As compared to T cells, B cell membrane are rich in which of the following antigen? Class 1, class 2, class 3 or HLA-B antigens?
Class 2.
Class 2. Okay, good. So B cell membranes are rich in class 2. Class 1 are mainly present on all T lymphocytes and macrophages, whereas class 3 is mainly complement components. Leukotrienes are secreted by, what are leukotrienes?
Macrophages.
Macrophages. Is that just a question to confuse you? Lymphokines are your interleukins. But leukotrienes are mainly produced by mast cells as a process of type 1 hypersensitivity or the any type of allergy. Okay. Which of the following anti-tumor compound is produced primarily by macrophages? Interferon, perforins, proteases, nitric oxide. Interferon are produced by which cells? Virally infected cells. Perforins are mainly produced by complements. The same way proteases are also mainly produced by complements. So it is the nitric acid which is mainly anti-tumor compound which is produced by the macrophages. T cell multiplication is stimulated by, this you should tell. Phytohemagglutinin. Okay, whereas bacterial endotoxin and Covan-1 strain will stimulate B cell, concanavalin A and phytohemagglutinin will stimulate T cells. Which of the following cells is the most susceptible to tolerance induction? What is tolerance? Tolerance is a state of unresponsiveness of the immune system to an antigen. Okay, of these cells, the B cells are the most, or the T helper cells are the most responsible for tolerance induction. And this tolerance may be based upon the type of antigen, like soluble antigens induce more amount of tolerance. The same way, the dose of antigen may be high dose or low dose antigen, which can induce a state of unresponsiveness of the immune system to the antigen which has entered the body. CD antigens are each expressed on only one cell type, are expressed on immune cells used by immunologists to mark them for separation, are found only on leukocytes, function as receptors for cytokines and CAM. Which is the answer? It is B, which is the expressed on immune cells, which are used by immunologists mainly for marking them as separation. They are called as markers, CD markers. Neonatal thymectomy leads to decreased size of germinal center, decreased size of paracortical area, increased antibody production by B cells, increased bone marrow production by lymphocytes. So thymus, which will it
Decrease
reduce
size
the
of paracortical areas.
decrease in paracortical area because it is a T cell dependent area which is present in the lymph nodes. Interferons activate B cells to make virus specific antibodies, are Th2 cytokines, are virus proteins that interfere with the activation of cytotoxic C cells, block virus infection of host cells.
C.
Which one? They block the virus infection. Interferons are produced by virally infected cells to inhibit the translation protein, TIP, translation inhibition protein. Okay. The antigens in thymus dependent humoral immune response show which of the following properties? They induce primarily IgM response, are mainly carbohydrate in nature, induce an active secondary response on booster injection, are exemplified by polymerized flagellin. So humoral immune response. They induce an active secondary immune response on booster injection. So there are two different types of antigens, thymus dependent and thymus independent antigens. The thymus independent antigens are structurally simple, whereas thymus dependent antigens are highly complex. And thymus independent antigens are, the immune response is dose dependent. The more the antigen, the more immune response. Whereas thymus dependent will be immunogenic over a wide range, and thymus independent antigens only IgM, whereas dependent will be isoclass switching to IgG. Independent antigens, no immunological memory, dependent will be immunological. For thymus independent antigens, you need not require any antigen processing, but for thymus dependent antigens, you need antigen processing. Examples are flagellin, capsular polysaccharides, and lipopolysaccharide of a gram-negative bacteria, whereas thymus dependent antigens are all serum proteins and erythrocytes. T cells are identified by answer. Which one? It is the rosette formation with sheep RBCs. EAC rosette is the erythrocyte complement rosette formation, which will occur only in case of B cells. Bacilli calmette guerin is sometimes used for passive immunization of tuberculosis, non-specific suppression of immune response, potentiation of humoral immune response, potentiation of cell-mediated immune response. Have you read in the papers that BCG vaccination can also be used as a treatment for COVID-19 infection? Why? Viruses which mediated immunity causes immunity, whether humoral or T cell. Viruses pa?
T cell.
It is T cell. So this can act as an adjuvant and they potentiate the cell mediated immune response. It is also called as an adjuvant. Okay. A 5-year-old child has been injured in a motor vehicle crash. To protect him from tetanus, a single booster dose injection of toxoid is given. Which of the following characteristic best describes the anamnestic response to the toxoid in comparison with the primary immune response? What is anamnestic response? Any idea? Secondary immune response is also called as an anamnestic response. So now you can answer. That period is longer after antigenic stimulus. More antibody is produced. IgM production predominates. More immunogen is required. Which one?
More antibodies produced.
Option
More antibody produced is the correct answer. Good. Interleukin 1 is produced by which cells? Macrophages. The injection of large doses of protein results in immune tolerance, and that is due to removal of antibody by excess antigen, catabolism of the antibody as rapidly as it is formed, production of a non-reacting antibody, suppression of B cells, T cells, or both. May be high dose tolerance and low dose tolerance. When there is a large dose of protein, which can, instead of stimulating an immune response, it may lead to tolerance. So suppression of B cells and T cells or both can occur, and that is called as high dose tolerance. Apart from T and B lymphocytes, the other class of lymphocyte is
NK cell.
NK cells. Good. NK cells provide immunity against
Innate immunity.
viruses. Cytokines are not able to inhibit the function of other cytokines, able to stimulate the synthesis of other cytokines produced by more than one cell type stored in the cell for quick release. It is not. It can inhibit and stimulate or not. It is not usually stored in the cell. It will be only produced instantaneously after an antigen stimulus. Okay. With regard to HLA class 1 antigen, all are true except they are expressed in all nucleated cells. They are essential for viral antigen recognition by cytotoxic cells. The genes for class 1 molecule are located in chromosome 6 and 15. CD4 positive cells only recognize antigens. With respect in association with HLA class 1 molecule. Which is the wrong answer? What is MHC restriction? MHC class 1 will attach only to CD8 positive T cells, and MHC class 2 will attach to CD4 positive T cells. Then which is wrong in this? So D is the wrong answer. With regard to interleukins, falses. Interleukin 2 is produced mainly by CD4 positive cells. Interleukin 3 stimulates the growth of hemopoietic stem cells. Interleukin 4 increases the production of interleukin 1. Interleukin 6 stimulates acute phase protein synthesis. So which is the false? This you have to read repeatedly and take notes. Only then you can. Interleukin 4 actually is the production of interleukin 1. It inhibits the production of interleukin 1. It does not increase the production of interleukin 1. The following are true about lymphocytes except T cells account for 20% of circulating lymphocytes. In spleen, B cells are found in the periarteriolar areas of the white pulp. In the lymph nodes, T cells occupy the paracortical area surrounding the germinal centers. B cells but not T cells express surface IgG. This is the wrong answer. B cells are mainly produced in the medullary cords of the spleen and not in the periarteriolar sheath, or the white pulp is usually the T cell dependent area of the spleen. Cells which have MHC class 2 are antigen presenting cell, cytotoxic T cells, target cells, none of the above. So all cells which have MHC class 2 are mainly the cytotoxic T cells or the CD8 positive T cells. Okay. Perforins are produced by suppressor T cells, memory helper T cells, plasma cells, NK cells. Which cells? It is the NK cells. Which of the following immunity deals with intracellular pathogens? Antibody mediated, cell mediated, both, none.
Cell mediated.
Huh?
Cell mediated.
Cell mediated. Can you give an example of an intracellular pathogen? Tuberculosis. Okay. MHC class 2 antigens are located in all of the following except dendritic cells, RBCs, T cells, B cells. This is the RBCs. Okay, it will be present. MHC class 2 antigens will be present on all the immune cells. The following are true about antigen presenting cell except Langerhans cells are the antigen presenting cells of the epidermis. Neutrophils are antigen presenting cells. CD8 cells recognize antigen presenting cells bearing MHC class 1. Tumor necrosis factor alpha can turn endothelial cells into an antigen presenting cells. So which is wrong? Neutrophils can never act as an antigen presenting cell. Okay. The following are true about interleukin 1 except produced by neutrophils, stimulates proliferation of T cells, acts on the hypothalamus to cause fever. It increases the production of prostaglandins by T cells. It is not produced by neutrophils. It is mainly produced by the macrophages. Okay. Secondary immune reaction is differentiated by primary by increase in time lag, decreased peak level of immunoglobulin, increased peak level of immunoglobulin, initial negative phase.
Increased peak level of immunoglobulin.
Increased peak level of immunoglobulin. Following are true about transfer factor. Transfer factor transmits the cell-mediated immunity. It is an extract from leukocytes. It transfers cell-mediated immunity. It promotes antibody synthesis. It stimulates release of lymphokines. which is not true. Transfer factor is mainly related to cell-mediated immunity. So it promotes antibody synthesis is the wrong answer. Which of the following immunity deals with extracellular pathogens? Antibody mediated, cell mediated, both, none. Intracellular, cell mediated. So extracellular is usually
Mediated.
antibody mediated. T helper cells
Antibody.
recognize class 1, class 2 processed antigen surface IgG. CD4 positive cells are T helper cells. They will recognize only MHC class 2. Class 1 will be recognized mainly by CD8 positive or the T cytotoxic T cells. Immunity can be acquired passively by catching a virus from a by shaking hands, receiving a vaccine of influenza virus grown in eggs, receiving serum from someone who has recovered from an infection, receiving leukocytes from an immune family member.
Receiving serum from a person who has recovered from the infection.
Okay, so with that we finish the immune response and structure and function of the immune system. These are 2 chapters. It is quite enormous, but we have to know the main points for you to attend the multiple choice questions which are usually asked in your examination. Okay, is it clear? Okay then, thank you.
Yes, ma'am. Clear. Thank you, ma'am.
Corner.